let's thank Gina once again. Thank you, very engaging talk, and thanks for all your questions. Now is my turn, and I'll sort of kind of introduce myself as I go. So my name is Anna. I'm an assistant professor at the Institute for Systems Biology and affiliate at University of Washington in Seattle. Cool. So Gina here did a great introduction into, why do single-cell transcriptomics in general? And by this point, it's been over a decade since we've had this ultra high throughput methods, like now it's 10x basically, that performs single-cell transcriptomics for all kinds of mammalian systems or eukaryotic systems, uncovers cell types and states and makes this atlases of different cell types. However, bacteria also have differentiated cell states or different gene expression cell states. And this is just a small example that I'm showing here. Of course, there may be a bunch of other different states in bacteria. But here are a few examples of a very well-known differentiated or distinct gene expression states in bacteria. Bacteria typically devote or very frequently devote just some subpopulation of themselves to produce matrix, to sporulate, to become competent for external DNA uptake and so on. And so how have we historically been studying the states is just looking at fluorescent reporters. Like this picture was from my graduate school where I studied sporulation and competence, and you can see them tagged with two fluorescent reporters, blue and red. Very quantitative, but also very low throughput. So what about transcriptional states in a much more complex microbial community, such as the microbiota, for example, right? It has hundreds of species. It may have non-culturable or non-modifiable members in which you just cannot stick a reporter in, right? Or even if you can, to some of them, your use of fluorescence is severely limited in anaerobic bacteria. And that's just one example, right, of challenges to measuring gene expression states in a single cell level in bacteria until this technology came about 5 years ago, right? If we can do single cell RNA sequencing in a similar fashion as for eukaryotic cells, but for bacteria, now we can address many different species. We can address complex samples because we do not need to modify them, right, to measure the gene expression states. We also don't need to select a set of probes. Based on our current knowledge or maybe some hypothesis, we can just do whole transcriptome profiling. So that's perfect, but as Gina mentioned, it's a very hard challenge. And basically how we came about to solve it is when I was a postdoc in Georg Selig lab at University of Washington, I developed one of the first methods to do so, and we called it microsplit, microbial split pool ligation transcriptomics. And I'm going to give you a short introduction of how it works mechanically, since we've got a few questions on that. So this is, and again, today we have also technologies that are adapted for droplet technologies. And I'm not going to talk about that today. Today I'm focusing on the split pool barcoding. So how it works is basically, even though it's a single cell technology, it never actually isolates single cells, right? So let that sink in, right? We never ever isolate single cells. We do not compartmentalize them into droplets or into microvels anywhere, right? Instead, we treat the cell itself as a container that holds its RNA molecules. Now, then we take the cell and we fix it with formaldehyde, which kind of locks the RNA molecules inside, and then we permeabilize the membrane and cell wall. Now that external enzymes and DNA barcodes can diffuse in and label the RNA molecules inside. Okay, so after this process, when we have fixed bacteria and permeabilized bacterial cells, we do split pool barcoding, and here's how it works again. The first split, we just transfer them to a 96-well plate, and at this point, each well here may hold hundreds of thousands of cells, right? It's not individual cell barcoding at this point. Every cell in a well will receive its own individual barcode. But again, every other cell in the same well will receive the same first barcode, right? So here we do reverse transcription in cells attaching barcode 1. But then when we pull the cells and mix them, we redistribute them randomly into a second set of 96 well barcodes, right? And so now imagine the cells that had the same first barcode that were neighbors in the first round. There is a very high chance statistically that they will end up in different wells in the second plate, right? And they will receive a different second barcode. So now we, just to get better statistics, we do it again. We do the third round, we mix the cells, we pull and redistribute them into a third set of wells. And after the third barcoding step, the cell, each RNA molecule in the cell will acquire a set of 3 individual barcodes that will label this, the RNA in the cell uniquely. And now we can pull, lyse, and sequence the libraries. And this is, again, how the numbers work by using 96 well plates in a 3 split pool barcoding routes. We generate 96 to the third unique barcodes, and then we use 24 forced barcode combinations just to label our sublibrary, which creates about 20 million unique barcode combinations. And if we are happy with 5% rate of doublets or lower, then we have a capacity to uniquely barcode 1 million cells. And all of that is done again on a benchtop. You do not need a device. You do not need to buy a commercial kit. All you need is basically a benchtop and a 12-channel pipette, so like that. Okay, so in our pilot publication, we profiled Bacillus subtilis cells. It's a model gram-positive organism. We grew it basically in a lab, sampled 10 optical density points across the growth curve. And what's also kind of great about this technology is that you can multiplex different samples in a single run. So all of these 10 different samples were multiplexed in a single experiment. And we retrieved about 25,000 single-cell transcriptomes mapped here on the UMAP, colored by the optical density of origin. And you can see that they cluster nicely corresponding to the growth stage. But we also see some discrete clusters that are irrespective of the growth stage, or maybe specific to several of them. For example, we see a prophage activation, right, in something like 36 cells out of 25,000, right? So this gives you an idea of sort of the resolution of this technology, of how small as a population can you find. And so that is that. This is our pilot publication. Since then, we've also published a detailed protocol in Nature Protocols, and I'll give you a link to that later if you want to try it out. And so in my lab at ISB, we basically have 2 goals. Our first objective is to continue developing tools for single-cell omics to study bacterial lifestyles. And beyond microsplit, we've also developed, or in the process of developing, a couple of other technologies based on it. One is single-cell meta-transcriptomics, or metasplit, which is optimized specifically for microbiome profiling, where you have a bunch of different species and strains with unknown composition. And the second one is mapsplit, which we use to do pooled genomic screens in bacteria using CRISPR. All right, and then we're also interested in applying these technologies to study bacterial biology. And some of the directions in my lab are studying biofilm development, looking at microbiome analysis. I'm not going to talk about this today, but feel free later if you have questions to come and talk to me. So today I'm going to present on a couple of projects in the lab. First is an example of how we use microsplit to study phage bacteria interactions. And the second one is on mapsplit, or pooled genomic screens to investigate gene regulatory networks in bacteria. All right, let's go on. Okay, we'll do questions later. So, so first of all, let's see what kind of biological questions beyond just studying subpopulations in bacteria per se, in cultured bacteria, can we envision for microsplit. And this is an example of where we can study interactions between 2 different entities. And specifically, we'll be looking at the lytic phages. Lytic bacteriophages are viruses that specifically infect and kill bacteria. And the gut is a source, a very rich source of diverse bacteriophages. In fact, this is the second most common entity in the gut microbiome after bacteria themselves. And they act as basically predators, right? They shape the composition and function of the microbiota. They're actually very important in controlling how our microbiome works. And there's a continuous arms race, right? Bacteria have defense mechanisms that protect them from these phages. And a few of them are listed here. Surface modifications or abortive infections, restriction modifications, etc., are all of those examples of these mechanisms. And so basically we asked, well, what is the role of single-cell heterogeneity and the pre-existing heterogeneity in bacteria in defending from lytic phages, specifically in the gut anaerobic species? And the secondary question that we actually were really interested in was whether we can use microsplit on gut anaerobic species. And the answer is yes, it works pretty well on Bacteroides. So this is the Bacteroides fragilis is our host. It's an anaerobic pathobiont of common in the gut. Pathobiont just means that it's pretty common in most people and it doesn't cause any harm normally, but only in case if the intestinal barrier is breached and if it escapes into the bloodstream, it can cause infections and actually quite serious, leading to abscesses and sepsis. So what we did, and this is a collaboration with Fred Hutch. The lead authors on this study are Annika and Dmitry from my lab, and then Norma is a postdoc at Fred Hutch. So Norma fished out a phage, a novel phage from Seattle wastewater, and characterized it and sequenced it. It's a lytic phage and is very specific to Bacteroides fragilis. And we grew B. fragilis on its own, and we grew B. fragilis that was treated with this phage at a very, very low multiplicity of infections. So basically, the cells could undergo a few rounds of division before the culture just collapsed, as you can see in the blue line. And before this culture collapsed, right, we were able to sample from it to do microsplit. So again, we have two samples here, bacteria that were grown alone versus bacteria that were grown with phage just before the lysis. And we've done microsplit on them. And on the left here, you see the UMAP, resulting UMAP, colored by the sample, right? So the orange cells are treated, which means they were grown with phage. The blue cells are untreated. And immediately you can see that, yes, the cells that were treated with phage and infected, presumably, form this sort of very discrete cluster on the right. However, we do see some treated cells clustered together with untreated, right? So which means a subpopulation of the sample was actually uninfected at the time when most of it was infected, and the culture, the bulk culture, looks like it's just going to crash and everything's going to die. But that's not true, in fact. Some cells remained uninfected and it survived. And indeed, if we look at where the phage reads mapped, we see that the cluster on the right is infected. And this is the distinctions, right? This is why we didn't call our samples infected or not infected. We call them treated and untreated. Now what can we do with this type of data? There are two types of analysis. One of them is called pseudotime analysis. So what we noticed is that the infected cluster sort of looked like it had a gradient of phage expression, right? And so we reasoned that because the cells had the time to undergo several cycles of division, it is likely that at the time of sampling, we caught cells at different stages of phage infection. And so now there is a computational technique that's called pseudotime trajectory analysis. And those of you who work with mammalian cells probably have seen it used on figuring out the differentiation trajectories, right, of mammalian cells. But here we used it to infer the infectious cycle of phage from a single time sampling, right? So how it works is basically it tries to arrange the cells along a trajectory based on the gradual shifts in gene expression of each individual cell, right? And so when we calculated this trajectory, we saw that the host and phage exhibit different patterns of gene expression along this pseudotime. That actually makes sense. And so if you look at the phage gene expression, for example, the early pseudotime corresponds to replication genes or DNA binding genes, for example, being expressed from the phage, while later pseudotime corresponds to structural gene expression, lysis, and capsid maturation genes. So overall, we were able to reconstruct the infection timeline from just a single sampling using this single cell data. So what else can we do with this data? We can look at the cells from the phage-treated sample that were not infected and ask what kind of protective genes were expressed in the cells that enabled them to survive phage attack. And so we did a lot of analysis here, and just going to sort of skim over them because of time. But in a nutshell, what we found is that the answer was the variable capsule types. So this Bacteroides fragilis have 8 types of capsules, PSA through PSH, and they're wired in a way that each cell can turn on just one type of capsule at a time, right? So in the population of cell, you will have all 8 very likely represented, and each cell only expresses a single type. And we were able to calculate the probability of infection for the cell expressing each capsule type. And interestingly, it varied from almost complete protection, 97% protected, for some capsule types like PSA and PSE, to only 14% chance of survival for the most protective capsules, actually. I think I had it backwards, right? So 97% of infection for some of the most vulnerable ones, and 14% of infection chance for the most protective ones. Interestingly, it's a spectrum. It's not just an on-off, right? So there are some complementary factors that are also important. And the complementary protection was provided by the fimbriae. So some of the clusters that we found in the uninfected subpopulations correspond to, again, stochastically switched on and off fimbriae by the genomic rearrangement of the promoters. And we found that cells, and apologies for that, I don't know what happened to the quality of this image, but what we found was basically the expression of fimbriae in the cells together with the most protective capsule provided almost complete protection from phage. And then finally, what about these phage defense systems that are so famous? Again, we found that their expression was heterogeneous in the population, and if on its own, the expression of any defense system was unable to protect the cells from phage. However, if the cells expressed the protective capsule type and several defense systems, it further protected them from phage almost completely. All right, so the conclusions from that is that pre-existing heterogeneity seems to enable Bacteroides fragilis to survive initial phage attack. And so the phage escape playbook for this particular species looks like basically a gamble, right? The whole population gambles of what kind of capsule am I going to produce? What kind of gene defense mechanism am I going to express? And then, depending on what kind of phage it encounters, some of the cells are protected from this phage, and then they can grow out and, then acquire more permanent genetic protection. For example, acquire CRISPR spacer or mutation. And then they can diverse phenotypes again and play this gamble again against the next phage. So that's that application. And then let me go and outline our next technology that we developed, which is called MapSplit. It's a pooled CRISPR screens for bacteria. Okay. So what are the gene regulatory networks in bacteria? And this is a little bit of an outdated graph. Okay, so it's for E. coli. And it just shows how all the transcription factors are connected to their regulons and what kind of genes do they activate, what kind of genes do they repress. And why do we want to map this, right? Because if you want to engineer bacteria for any kind of maybe metabolic engineering, we need to rewire these pathways and we need to understand how they connect to each other. For infectious disease as well, right? We need to know what kind of networks are involved in, for example, antimicrobial resistance. So these are just 2 examples, but overall gene regulatory networks are really useful to know for many applications in microbiology. So how normally people do it is they basically do bulk RNA-seq on hundreds and hundreds of different conditions. You grow cells in different media with different stressors, and then you map the gene expression responses. But this is low throughput and pretty ineffective and also very costly. So enter high content pooled CRISPR screens. And you know this technology from mammalian cells, it's called Perturb-seq or crop-seq. right? And it's been about a decade since the first papers came out of Perturb-seq in 2016 and crop-seq in 2017. And basically what they do is they make this sort of CRISPR perturbations that switch off or downregulate expression of a specific target gene. And in the mammalian case, they make them, package them in lentiviruses, and they transfect the eukaryotic cells and then do droplet RNA-seq. The important part here is that basically together with each perturbation, you include a transcript. And the transcript is your barcode. It encodes the identity of the perturbation. And then when you do RNA-seq in single cells, you can basically read out both the transcription profile of the perturbation and this barcode that shows you the identity of the perturbation. So you connect the target with its response. And so now basically, a decade later, we introduced this method for bacteria. A similar method that we called map split is mapping annotations for, I forget actually what it stands for, sorry. But it's called map split, and it's a high content pooled CRISPR screens in bacteria. And so instead of the lentiviral pool, we just make a plasmid pool for different CRISPR perturbations. It can be inhibition or activation. We use it on bacteria. We just transform bacteria with a plasmid pool, and then we do microsplit instead of droplet transcriptomics. Again, how it works, you build single guide RNA library for either interference or activation of target transcription factor that you'd like, or any gene really for genomic screens. You transform cells with your coupled single guide RNA and guide barcode construct, which is essentially your transcript, right? That tells you what kind of perturbation do you do for this cell. And then you perform microsplit that links transcriptome and perturbation identity. All right, cool. So what we've done with it, we've done a bunch of different things, including studying gene interactions. But I'm going to skim over all that and just show you this, our biggest experiment that profiles 118 different pooled perturbations, 36 activating and 82 inhibition, targeting 52 transcription factors of interest, and it's a mix of well-known transcription factors for validation of technology, as well as unknown transcription factors. And believe it or not, E. coli actually still has a lot of uncharacterized transcription factors. And we were able to map responses of this partially characterized and unknown transcription factors on the right here, and we were also able to both validate our technology and also discover new regulations, even for transcription factors that were characterized before. So we were able overall to add 118 interactions and 19 transcription factors of our library. And what's also interesting is that we get single cell data. So now we can ask questions like, if we perturb this transcription factor, right, if we downregulate it, is the response the same in all cells? Or do we have a subpopulation of cells responding differently, right? And so we can, if we cluster all the cells in our library and then map the perturbations onto this sort of UMAP, we can see convergent and divergent phenotypes. And what I mean by that is basically you can have the result of one perturbation diverge into two different transcriptional responses, or three in some cases. And a lot of them are actually resulting in differentially expressed motility genes, what's interesting, and also carbon metabolism. So these two phenotypes seem to be connected to a lot of different transcription factors. And vice versa, right? Sometimes you can get the same output from multiple different perturbations. And what is one example of what we can do is this gene regulatory mapping tool, MapSplit, is, for example, use it for bioproduction, right? To figure out how do we, what kind of perturbations to the native transcription factors will enable us to engineer the strains, insert the bioorthogonal reactions there for basically producing, for bioproduction of different materials, right? And so to recap, the field of bacterial single-cell transcriptomics is very new. The first publication for high-throughput bacterial single-cell RNA-seq came out just about 5 years ago. But since then, we've had an explosion of different techniques. There's now at least 5 or 6 techniques, a few of them split pool barcoding based, and another bunch of them droplet-based, including 10x-based and also custom microfluidic devices-based. There's one probe-based technique as well called proboxic. So there's a lot of techniques now to choose from, and microsplit is just one of them that we are developing into more targeted applications and technologies, for example, for microbiome profiling or for pooled genomic screens. And I don't think this is the end of it. I think this is just the beginning of the explosion in this field. And some helpful resources if you'd like to try it in your lab. I think the one that's on your top left is probably the most important. Is the microsplit protocol that we published as a protocol again, and it has a troubleshooting guide. It tells you how to optimize it for different microbial species of interest, etc. And then, of course, feel free to send us a note if you'd like to try it, and maybe we can think about your application with you. And also, here are the couple links to our map split technology and to microsplit original publication. Cool. With that, I'm thanking my lab. These are lab members on the left, and especially Dmitry and Annika, who were the lead authors of the phage paper, and also Jacob and Norma. All right. Thanks. Happy to take any questions now.
Excellent technology. I still split and pull, right? 3 round, and then you start to do the lysis and the sequence, right? I like to know this technology. You can get enough number of the cells, but for how many transcripts you can get per cell?
Yes, excellent question. So resolution is what we call it, and the resolution can vary, and there's multiple factors. I think the biggest one is basically species to species variation in how many transcripts intrinsically are there in a single microbial cell. And what's kind of surprising, what was surprising to us, is that it's not actually very trivial to answer that question. The field has not really looked at that very much. And there's a huge variation, right? And some bacteria that are model species frequently grown in the lab do not actually have that many transcripts per cell intrinsically. It seems like there's a very high turnover. So if you are sampling at just one time point, most of the cells don't have many transcripts. But in general, we can capture tens to hundreds of transcripts per cell. The best performers are probably like 300 to 400 transcripts per cell, depending on the growth stage.
Hi, thanks for the talk. Have you looked at population diversity and expression in some of these systems? I know, so I come from more of a background of microbiome and pathogen detection, and thinking about it from the perspective of microbiome health, could you use this type of approach to look at the health of a microbiome and maybe use it to identify transcript markers and things like that that might be teetering or leading to the edge of a dysbiosis system or something like that?
Yes, absolutely. And I think this is where the field is going. Again, like Gina mentioned, there were a couple of papers that came out. One on the oral microbiome just came out, and we are working on the gut microbiome application, which is very hard because it's so diverse. But yes, absolutely. I think this is a very ripe sort of application for this technology. And so far, I have not seen too much exploration of a diseased microbiome state with this technology. But again, usually we pilot with healthy microbiome, right, just to see what's there. What is the baseline? And the baseline is amazingly diverse, as you know, between individuals even. So it will be a little bit hard to compare, especially across studies. But also, if you just take into account the inter-individual variation, and if you now look closer in the individual microbe level, there's even more variation. But I think we do see patterns, and this is just a preview of, I have not talked about it at all, but we do see what we think of are close to universal patterns, global patterns of gene expression that we see. And perhaps these are the patterns that will change in health and disease that we could, probably measure and compare across studies. And so what I mean by the global pattern is, for example, stress response genes, right? Typically, we do in every species that we look at single cell with single cell resolution, we see some kind of a stress cluster, right? So to speak, right? Some kind of stress response. It could be different, but it seems to be a generalizable feature, and we do see more of those come up in the microbiome data than in isolated strains. So we can see how these clusters will change, right? In disease, maybe there's some dysbiosis and the community composition change, right? That will drive this patterns change.
Thank you.
Right?
So I think, thank you, great talk. Both of these talks were awesome, thank you. In our university, we have an investigator that's looking at the role of bacterial microbiome in the gut and its association with colorectal cancer. And then also looking at ways to modulate microbiomes that are progressing towards a more cancer-related state to basically repopulate the gut with, with good bacteria and, get away from the organisms that are, causing or responsible for some of the disease or the state of disease. I don't think it's one bacteria that's, it's a consortium. But they tend to look at it from the point of view of the human and not necessarily like how are these bacteria changing the human cells versus how is this microbiome transcript and the way that they're expressing and the way that they're coming together, how are they actually influencing this instead of, because bacteria, you have the different serotypes, and so you have different things that are happening. So it may not just be as simple as, oh, well, we have the presence of this bacteria and these 10 bacteria, right? But it could be like, okay, these are the serotypes actually that have to be considered instead of just.
Right.
Anyways.
So there's definitely multiple layers of diversity
Right?
in the microbiome, and that's been the common theme, right? So it's not just the composition that changes. Inside the composition change, there is a functional change. The bacteria change the gene expression. There's different strains that can change gene expression differently between strains, but that's still genetic. And if you go now into the sort of inter-individual variation in gene expression, then even within those strains, you can see further variation. So it's like a Russian nesting doll, right? It's one variation inside the other variation, and you go down to the very bottom of it, right? With that single cell transcriptomics. So this is, I think, it's very hard. It's a very hard bioinformatics problem to disentangle all of these layers. But yes, this is, I think, where we're going with this.
Yeah, I think you're going to have to change the paradigm because whenever you see these microbiology, microbiome talks, generally, they're talking about an organism, but they're only looking at the 16S. They're not looking at the changes. And anyways, this is just fantastic. And I think it's going to change the way we do science in cancer and in other areas. So anyways, I'm just very excited.
Thank you. Okay, so we can, I think, now just conclude the session, and you guys are free to go and enjoy your lunch, but if you want to talk, please, we'll hang around.
For those who are staying for the DEIB session, lunch is outside. For those who want to go to the library talk or other activities, it's in the go to their exhibit hall. So you're welcome to stay and have the lunch just outside. Thank you. Hello, everyone. I hope you are enjoying your lunch. When you're having lunch, I just want to say quickly, each table, or if you do not have, I'll move one, have this. So mostly are empty. And then if you, among whoever are already on the table, you can determine a topic to stimulate discussion. And for later comers, they could join your table. So this is the example of that, this table, and come up with rewards and recognition. And then we just have one pen. So I'm going to pass it around. This one pen, you can write on that. Thank you. Enjoy your lunch. And during the same time, you discuss. So Mary will have some slides just to show briefly during lunch. Thank you.